Much for the invitation. It's great to be in Marseille, even if virtually. Today I'll be talking about the noradrenergic system in aging. I want to give you a big picture of why the locus coeruleus matters and how its anatomy and function relate to aging and cognition, and how it also makes it vulnerable to changes in Alzheimer's disease and degeneration in aging, and then turn to whether we can actually intervene to optimize locus coeruleus function. So the locus coeruleus is this small brainstem nucleus that's the source of most of the brain's noradrenaline, and it influences many different regions of the brain. It has many extensive routes through which it releases noradrenaline, and it's composed of neurons that have long axons that in order to be able to do this. And while it innervates many different regions of the brain, its inputs come from a more limited set of brain regions, and these can be thought of as all relating to some sort of arousal, whether it's related to sleep, alertness, motivation, stress, threat in the amygdala, or even cognitive effort in the prefrontal cortex. So you think about working memory or trying to do a cognitively challenging studies way back many years ago by Danny Kahneman, where they looked at pupil dilation when people were trying to remember lists of digits, and the longer the load, the greater the pupil gets. It's just a beautiful effect. And the pupil is, of course, affected by the locus coeruleus. So as locus coeruleus activity increases, pupils dilate. So that's one window into the locus coeruleus. So we have this system that can be thought of as an arousal hub region that gets these inputs from these various regions, integrates them, and sends out these signals when the system is aroused, when it needs energy, when it needs to focus. And, some years ago, it used to be thought about as a broadcast system that would broadly send this message out. And one of the ways that fits with this idea is the anatomy fits with this idea is that locus coeruleus axons generally release noradrenaline at what are called varicosities. So here these arrows point to these what look like beads on a necklace. And you can see this would be a large varicosity. Here's a medium one. Here's a really small one down here. And this is the standard, the default way that the locus coeruleus releases noradrenaline instead of doing it in synapses. And there's another really interesting part about the locus coeruleus anatomy, which is that the axons from these noradrenergic neurons are very long, as I mentioned, in order to be able to influence so much activity throughout cortex. But they have very little myelin, which is surprising if you think about it. Myelin is this amazing insulation that allows the neural transmission to be faster in long neurons. So why is it that this system that has such long neurons, and one would think would like to send quick messages if it's the arousal hub region, why is it foregoing this, great possible advantage in terms of myelin? And it seems that probably one disadvantage of myelin is that it isolates the axon from its surroundings. So without myelin, this axon can communicate better with the conditions that are around it. And this property of being able to interact with its surrounding is a key part of a theory we developed called the gain model. And this theory was an answer to a question that I developed from trying to understand how emotional arousal affects memory and attention. And, as a researcher interested in emotion, one of the primary observations is that under emotion, we end up really focused on some things and ignoring others. We end up having very vivid memories, but these memories often have really salient gaps. So my favorite example is a memory from a friend of mine who she worked for a national company that every year had a large meeting where hundreds of people would gather in an auditorium, and every year they had this award that they would give out via with a fun method where they would interview people and keep it secret who the person winning the award was in the video until the very end. And she was watching this video this year and one year and couldn't figure out who the person was, but figured she really should know the person because they were working on very similar things to her. And then they said this person was a shot putter. And a shot putting is a very obscure track event. And she is a shot putter. And so she suddenly knew they were talking about her. And she remembers that moment really vividly, but she doesn't remember how she got up on stage, what she said. And she later got the opportunity to watch the video, and she was shocked at how little she had remembered of the rest of the video. So this illustrates that sort of dichotomy that emotional arousal can have at the very same event, same time frame. It can be enhancing memory and attention to some things that are really stand out and other things not at all. And so in the lab, we had realized that we could predict what arousal would enhance and what it would impair. It would enhance the things that are more salient and impair the things that are less salient. But I couldn't understand how this could work in the brain. How can noradrenaline, which is known to be associated with the enhancements in memory for emotional things, how could it be having such different effects on things that are highly arousing versus things that are not? And so I brought my problem to Carolyn Harley, who had spent her career working on the neurophysiology of noradrenaline in the hippocampus. And we joined with two of my lab members, Michiko Sakaki and Dave Cluett, and we worked on this problem, trying to think about how this could happen. And the key insight we had is that interactions between glutamate and the release of noradrenaline could lead to hotspots of really increased glutamatergic activity. And the way this would work is that this is a cartoon of a noradrenergic axon with those varicosities along the way. And this is a synapse that's somewhere in the vicinity. And a glutamatergic synapse releases glutamate for these two neurons to communicate with each other. But not all the glutamate stays in the synapse, and some of it leaks out and influences activity release of noradrenaline at those varicosities. And this has been well documented that glutamate influences the release of noradrenaline at these varicosities. So the way that works is that glutamate stimulates NMDA receptors here that have a special property that the neuron itself has to be depolarized or activated in order for that glutamate to actually do anything at the receptor. And if those two things are true, that there's glutamate activating it and the neuron is depolarized, then it leads to noradrenaline release. And that noradrenaline release can influence beta adrenergic receptors at this glutamatergic synapse. Beta adrenergic receptors are a less used adrenergic receptor type in the brain. The workhorse of adrenergic receptors in the brain are alpha-2A receptors. These receptors are generally inhibitory and they have a low threshold for action. So it's very interesting, again, counterintuitive. If you look at animals and you take them from rest to higher levels of activity, what you see in the brain is that neurons become quieter as this arousal level of the animal is increasing. And that global effect of inhibiting neural activity is due to these alpha-2A receptors. So as the locus coeruleus is becoming tonically more active and it's releasing a little bit more noradrenaline throughout the cortex, it's going to inhibit activity throughout the cortex. But the beta adrenergic receptors require about 30 times more noradrenaline to be activated. They're quite a high threshold of activation. And so in order to activate those beta adrenergic receptors, you need a lot more noradrenaline, which is exactly what these hotspots can do, because the glutamate is going to lead to relatively more noradrenaline being released here than elsewhere, enough that it could potentially activate these beta adrenergic receptors that are excitatory, lead to more glutamate release. And also, these beta adrenergic receptors are triggers for synaptic plasticity. So they release plasticity-related proteins. They lead to the release of plasticity-related proteins. And those help lead to synaptic change that helps consolidate memories. So we have here a way that the noradrenergic system can have very different effects depending on what's going on at those specific neurons. So at networks where we have high levels of glutamatergic activity, we are going to see relatively more noradrenaline release, and that's going to lead to this hotspot of activity. So these properties of the locus coeruleus neurons are shared actually by other neurotransmitter systems. So dopamine, serotonin, and acetylcholine all have this property of being having long and poorly myelinated neurons. And as I mentioned, this has this advantage of allowing effective communication with the milieu around the axons. They also all are small nuclei that are highly connected with other brain regions. And this is great for having a big outsized influence in the brain. And on the other hand, both of these things lead to some risk in aging and neurodegenerative diseases. So as you probably know, neurodegenerative diseases have the hallmark that proteins that otherwise would just be waste products swept out of the brain aggregate dysfunctionally. So in a negative way, they aggregate and disrupt function of neurons. And in particular, Brock, who I would say is the genius of postmortem analysis of brains, he's looked at many, many thousands of brains of different neurodegenerative diseases and healthy people. And what he observed was that it is the long, poorly myelinated neurons that show protein aggregation in neurodegenerative diseases. So that's one risk factor. And then another is that protein aggregation, these neurodegenerative diseases have a prion-like spread through networks. And so the more of a network hub a brain region is, the more vulnerable it is to this spread of pathology. No matter where the pathology starts, it's just more likely to hit a very well-connected brain region. And in particular, the locus coeruleus seems especially vulnerable to Alzheimer's disease. So it can be considered perhaps ground zero of Alzheimer's disease based on, in particular, these striking postmortem findings from Brock and colleagues. So they had over 2,000 autopsy brains from age 1 to 100, and they looked at whether there was the very initial signs of Alzheimer's disease. But in this study, they noted that you never see those tangles in transentorhinal cortex if you don't also see hyperphosphorylated, or what could be called persistently phosphorylated tau, in the locus coeruleus. So when the tau gets phosphorylated, hyperphosphorylated, it no longer has this magnetic charge that makes it can stick to the microtubules and axons. And it seems that then it's primed, it's more likely to form these tangles. And they took any brain they could get, basically, as I read it. They said non-selected cases from the mortuary. So you have many more that are in their 60s and many fewer in the kids. But, of the kids, most of them didn't have any pathology, but a few did. And by adolescence, most of the brains were showing at least hyperphosphorylated tau in the locus coeruleus. And by the 100 brains that they had between age 31 and 40, all 100 had hyperphosphorylated tau in the locus coeruleus, and close to 30% had what would be considered stage 1 Alzheimer's disease, or these tau tangles in transentorhinal cortex. And this just keeps marching on, and I don't show the later stages. This is just showing the percent of cases that would have at least stage 1 Alzheimer's disease. And it, of course, increases and spreads through the cortex as the stages continue. So this really leads one to think the LC is really important, and that things are going on in the LC many years before the development of Alzheimer's disease. However, until recently, the locus coeruleus was relatively ignored in aging research, and this is mainly because of how hard it was to measure in vivo. On typical MRI scans, there's no landmarks for the LC, and it really wasn't possible to measure it. So the question that we for years haven't really even paid attention to is whether it's important for maintaining cognition in late life. And so we have recently had the advantage of a new MRI sequence that was developed that detects high contrast at the locus coeruleus. So this high contrast is not yet known exactly what biologically is leading to it. It was assumed that it was neuromelanin, and it was validated post-mortem that the location of that contrast matches with neuromelanin. So neuromelanin is a pigmented polymer that's a byproduct of catecholamine metabolism. And so, of course, noradrenaline being a catecholamine, there's a lot of neuromelanin is formed in the locus coeruleus, also in the substantia nigra BTA regions due to dopamine. And so this is actually called neuromelanin imaging quite often nowadays, but I think that we will find that that's a misnomer. There's recent evidence that animals without neuromelanin also show this contrast, and physicists have been doing imaging and showing that at physiological levels, neuromelanin doesn't lead to this contrast. Instead, it seems to be related to magnetization transfer effects that are related to water content in molecules. And the size of those neuromelanin neurons are really large compared to other neurons. And so fluid dynamics could be quite different. So we have a lot to learn, but in terms of this signal, it's proven to be quite specific to noradrenaline-containing neurons in the locus coeruleus. And we were the first to look at how it might relate to cognition in healthy older adults, and we found a relationship. We were using data from a study that wasn't designed to look at this, so we had relatively crude measures, but we looked at a cognitive reserve score that we put together and found that older adults with higher LC-MRI contrast had higher scores on this cognitive reserve, and in particular, they had higher verbal IQ. And then after that, Martin Dahl, now a research scientist at the Max Planck, started working on looking at this in the base 2 sample. I was on sabbatical in Berlin for a year, and at that point we discussed and ended up putting in our sequence, the sequence that we'd used in LA, we adapted for, Martin adapted for the base 2 sample, added it to this relatively large sample, and Martin developed a semi-automatic method to be able to detect the locus coeruleus contrast in these images, and found that the rostral LC contrast was significantly associated with episodic memory performance. And since then, we've revisited this sample, realizing that there were some other scans in there, the magnetization transfer scans that had already been in the data collection protocol because other researchers were interested in the dopaminergic contributions to aging. Those sequences actually also show contrast at the locus coeruleus. So now in this sample, we have actually, if we take 2 of the magnetization transfer scans, we have 3 different signals of the locus coeruleus. And we also have the substantia nigra ventral tegmental area contrast that is indicated by these magnetization transfer scans. The other scan, the field of view isn't high enough for us to capture these regions. And so we have this opportunity here to compare dopaminergic and noradrenergic brain regions and how they contribute, are related to cognition and aging. And in particular, in this sample, we have really good measures of working memory and episodic memory. And as it turns out, these are very interesting in the context of noradrenaline and dopamine. So both of noradrenaline and dopamine influence hippocampal function. And the locus coeruleus has these well-documented projections via the dorsal noradrenergic ascending bundle to the hippocampus. And this is an image just showing how we can actually detect this in diffusion imaging scans looking at white matter. And for decades, it was assumed that the ventral tegmental area was the source of hippocampal dopamine. And there were a lot of models about dopaminergic influences to memory because it was documented that dopamine was having an influence on episodic memory, but it wasn't clear where that dopamine was actually coming from. And this was discovered not too long ago, that actually most of the dopamine in the hippocampus is released by the locus coeruleus. It was a big surprise. And so the locus coeruleus seems to have a much stronger influence over hippocampal function than dopamine does, is the current understanding. And so we would expect in our sample that the locus coeruleus might be more strongly related to episodic memory than the dopaminergic BTA. On the other hand, prefrontal working memory depends quite strongly on dopamine. So these are some really nice, beautiful findings from Goldman-Rakesh and her colleagues from many years ago. So these data show basically the white dots are controls, and this is working memory performance. Here, this is ablation of the prefrontal cortex. So no surprise, ablation of the prefrontal cortex, shown here with the black dots, leads to worse working memory performance than if a monkey has prefrontal cortex. But interestingly, they looked at temporarily depleting dopamine. And you can see that that has almost the same impact as ablating the prefrontal cortex in terms of working memory. And on the other hand, noradrenaline, depriving the prefrontal cortex of noradrenaline, didn't have much of an effect, and serotonin also. And these sort of data supporting the key role of dopamine have been there's been a lot of data since then. There's also links between noradrenaline and working memory, but they're not as strong. And so here we might expect the ventral tegmental dopaminergic regions to be more associated with working memory in our sample. And that's indeed what we found. I know that this is, whoops, hard to see, but we used some structural equation modeling where we had these multiple measures of working memory, multiple measures of episodic memory, and looked at how they related to these multiple measures of the locus coeruleus and multiple measures of these dopaminergic ventral tegmental substantia nigra regions. And what we found is really interesting. The locus coeruleus was related to episodic memory when we controlled for the influences of the substantia nigra VTA region, and vice versa, the SNVTA was related to working memory when we controlled for the other influences. And the medial temporal lobe, which of course is strongly associated with memory, we examined how the structure of the medial temporal lobe, whether that would account for the findings that we had with the locus coeruleus. And the locus coeruleus is still showing variance associated with episodic memory, even when we account for the medial temporal lobe structural variability. So it seems that the locus coeruleus is associated with episodic memory in late life. There's a lot of other evidence showing how important it is in neuroplasticity, brain changes during novelty, new learning, also, as I mentioned, emotional experiences. So it's a really important brain region for learning and memory in particular, also attention as well. Yet it's the first place that Alzheimer's pathology is detected. And so the big question for us is whether we can do something about this. Can we actually intervene to optimize locus coeruleus function? And here, a first important question is, what would be the goal of the intervention? Should we be aiming to increase locus coeruleus activity? And I would say strongly, no, this would be a bad idea to just overall increase locus coeruleus activity in the aging brain. And here's one reason. So you might have heard about the glymphatic system. This is a brain waste clearance system that is the analogy of the lymphatic system, and it involves glial cells, which is why it's called the glymphatic system. And those glial cells form an outer cylinder around blood vessels. So you've got the artery here, and you have CSF that is flowing around that artery that can seep out into those interstitial spaces between brain cells via water channels in those glial cells. And the idea that Maiken Nedergaard and her colleagues have been developing for over 10 years is that these fluid-filled passages through the brain are really important for sweeping out brain waste. And this happens in particular during sleep. And one reason this happens in particular during sleep is the role of noradrenaline. So Nedergaard and her colleagues have administered cocktails of noradrenergic agonists, and they found that those lead to swelling of the brain cells that in turn reduces that fluid-filled space between the cells, reducing the ability for flow to be able to happen. And so on the other hand, when they administer to TBI, a traumatic brain injury model rodent, where lymphatic flow decreases quite dramatically, if they administer a cocktail of noradrenergic antagonists, that flow improves, those fluid-filled spaces become larger. So noradrenaline is modulating how effectively this waste clearance system works. And so it doesn't work very well during high noradrenergic times of the day when people are awake and active, and it works much better when people are asleep and there's low levels of noradrenaline. So we obviously don't want to have something that's just going to be tonically increasing noradrenergic activity. And another really interesting thing that's important to understand is that there are two sort of two basic patterns of locus coeruleus activity. So one is just tonic level of firing that's ongoing, and another would be phasic, large bursts of activity. And those have been associated with different states, with the positive state with novelty. It's associated with new learning and with being able to consolidate and make these new memories, which might be in part because those beta adrenergic receptors are going to be more likely to be activated when you have a large burst of noradrenaline. And on the other hand, tonic firing of noradrenaline is associated with anxiety and a stress-like state. And here, this is work by a group in Newfoundland that included Carolyn Harley. And here what they did was they developed a rat model where they could optogenetically stimulate the locus coeruleus, so they could activate the locus coeruleus in whatever pattern they wanted. And when they activated it in a phasic pattern, the rodent explored more, which would indicate less stress. And when they activated it in a tonic mode, it froze. It was more likely to freeze, indicating anxiety. So the next thing these researchers did, which is really an amazing series of steps in this process, they first took rodents and created a rat model of this Brock stages of abnormal tau. And, tau tangles are not a feature of the normal rodent brain. So they inserted hyperphosphorylated tau from humans into the rat locus coeruleus and over months observed what happened. And they found that the rodents with this hyperphosphorylated tau showed the sort of progression that would be expected from the human postmortem literature. Slowly, the pathology spread along the ascending tracks and eventually reached the olfactory bulb, which is in the medial temporal region, and the rats developed olfactory memory deficits, and that's shown here. This is a control rat that's doing a difficult olfactory discrimination task. It's doing pretty well. The one that months ago had the abnormal tau inserted into its locus coeruleus is really falling apart on this memory task. Now, the really exciting part is that a second thing that they did was they added this ability to optogenetically stimulate the locus coeruleus. And they found that if they stimulated the locus coeruleus in a phasic pattern for daily sessions for weeks, that rescued, they were doing this during the same time period that these, in their previous study, they'd found this collapse of memory. This rescued this memory performance, whereas the tonic stimulation, if anything, made it worse. So I think these findings are really important for us to understand and pay attention to, because they indicate the locus coeruleus is a complex region that has both positive effects, negative effects. It has different modes of action, and that those matter for whether something is going to be beneficial in aging or not. And if we're thinking about this from an intervention perspective, I think the first place to start is to try to reduce tonic levels of LC activity in older adults. And the reason is that tonic and phasic activity are highly anti-correlated. So when there's high tonic levels of LC activity, there's not much scope for a high phasic response, and you don't see much of a phasic response. And this is really beautifully illustrated here by findings looking at, first, what's tonic LC activity look like? It's higher when the animal is awake and active. Quiet wait goes down, non-REM sleep, it goes down further. The really striking findings, and to me, just really surprising, are that the biggest locus coeruleus response, phasic response to a loud noise is actually during sleep. So this, follows this relationship better than any sort of, waking awareness or any other sort of processes that might orient it more to noise. And so this matters in aging because we don't know yet, but the evidence so far suggests that older adults probably have hyperactive locus coeruleus the same way that they have hyperactive noradrenergic activity systemically. So you see high hyperactive sympathetic nerve activity. That's noradrenergic activity in peripheral nerves in the body. You see higher noradrenaline in blood and in tissue. You see higher noradrenaline also in CSF, cerebral spinal fluid, which would indicate brain source of that noradrenaline. We don't yet have any way to measure tonic activity of the locus coeruleus in living humans. And so we don't know, but I would, right now, my hypothesis is that there's hyperactive locus coeruleus in older adults. And so if we want to see those benefits of phasic activity, like when they're actually doing something novel and interesting, those benefits are going to accrue most when the locus coeruleus is not in a hyperactive tonic state. And this is generally a really striking aspect of the aging physiology, is this difference between the noradrenergic / sympathetic system and it being hyperactive and the parasympathetic system. So in younger adults, the noradrenergic system or sympathetic activity is quite high during the day and it's quite low during the night. And this is beneficial because we need that noradrenergic activity to learn new things, to be active, to be able to deal with challenges. And on the other hand, we need the low noradrenergic activity during night so we can do things like waste clearance. And the parasympathetic system also contributes quite a bit to that rest, digest, restorative aspect of sleep. And parasympathetic system is usually lower, less active during the day. In older adults, this pattern is disrupted in that they show hyperactive noradrenergic sympathetic activity in the periphery that's higher than younger adults in the daytime and doesn't decrease as much during the night. And on the other hand, the parasympathetic system in aging is basically almost starting to just collapse. It's really low during the day and it doesn't increase very much during the night. And this is beautifully illustrated here for the parasympathetic system with data from over 900 people, where they looked at heart rate variability, which is the primary measure we use of the parasympathetic system. And I'll get into that a little bit more in a few minutes. The drop is quite striking. So these are 18 to 24-year-olds who are showing this circadian rhythm that's quite strong and also higher overall heart rate variability compared to those in their 50s and 60s. And this, if you want percentages, it's really a huge difference. The, Fitbit is one device that a lot of people wear and measures heart rate. They estimated that there was a study with 8 or 9 million Fitbit users that was published recently that estimated that between age 20 and 60, heart rate variability drops by 80%. And this has been corroborated by longitudinal studies with hundreds of people where it's dropping precipitously in midlife. So for instance, in the decades in one's 40s, it's dropping by 50% on average. So huge decreases in the parasympathetic activity with aging. So one way that we might be able to intervene is to stimulate the blood pressure feedback system. So this is a system in which, when you stand up, your blood shifts around and blood pressure changes, and you might feel faint for a moment. That is your blood pressure system not yet having this feedback loop complete in order to adjust for changes in pressure. And what happens in the baroreflex is that there are mechanical receptors on the carotid sinus and also the aortic arch of the heart that sense stretch. And stretch is, of course, indicating more blood in the vessels, more blood pressure. And so those mechanical receptors send messages to the brainstem, and in particular to the nucleus tractus solitarius, that then, as part of this feedback loop, increases parasympathetic suppression of the speed of your heart. So it slows down the heart, which of course, if you slow down, you're going to have fewer beats per minute, less blood going into vessels. And then it also triggers, suppresses the sympathetic activity that would otherwise constrict vessels. So it's going to, again, reduce this blood pressure. And what's shown here is a nice diagram of this from Wikipedia, where they were showing that there are actually therapies where they implant devices and they mechanically simulate that there's pressure at this, the mechanical receptors are going to feel the pressure here, so that they will send this signal to the brain and initiate this loop. And that has been shown to reduce blood pressure. Now, this feedback loop takes a few seconds to complete, leading to an oscillation in heart rate of about 10 seconds. And for us, importantly, animal studies have shown that baroreflex stimulation reduces tonic LC noradrenergic activity. So if we can somehow harness this loop and stimulate it, we might be able to reduce older adults' tonic LC noradrenergic activity. So first, just how we might be able to do that, I want to show you that heart rate is very strongly influenced by breathing. So these are data from 4 of our participants in our study, one of our studies, and they're just sitting and resting. And what you can see is that they're showing increases in heart rate as they inhale and decreases in heart rate as they exhale. And this is just plotting heart rate over time. And so you can view basically heart rate variability plotted over time. And, a lot of consumer devices now are showing you your heart rate. There's a lot of attention on heart rate variability. I just wanted to see your intuition as to which of these is showing the highest heart rate variability. And so I believe Anne will be able to launch a quiz where you can pick A, B, C, or D. And just pick one. If you've already seen this from one of if you've seen another talk of mine, I have presented this before, don't answer. But if this is the first time seeing these data, please make your best guess as to which one has the highest heart rate variability. And I will specify high frequency heart rate variability, which is one of the most commonly used measures of heart rate variability. So, Anne, maybe you can keep an eye. I don't actually see the tally, but maybe you can see when most people have answered.
Yes, for now, like half of the people answered, so maybe we can wait a few more minutes, a few more seconds.
Great. Terrific. So you can see the answers here. The most popular answer was D, and the least popular answer was A. And this is a typical response, given this. This is just real data. I wasn't setting this up to try to fool anyone, but I think heart rate variability is a misnomer, and people have the wrong intuition about what it looks like. And so actually, the real results are the opposite of intuition. So this person, A, who looks regular as a metronome, heart rate's going up every time they breathe, down every time they inhale, down every time they exhale, nothing else really is happening. They have the highest high frequency heart rate variability. And D, the most popular answer, actually has the lowest. And this ranking is similar if we look at RMSSD, which is a time domain measure of heart rate variability. So why is this intuition so wrong? What we're really looking at when we're looking at high frequency heart rate variability is that response to breathing, and in particular, the power at the frequency of breathing. If you're breathing regularly and you're showing a large oscillation, you're going to have a very large peak and a lot of power, which is what increases that value. Low frequency heart rate variability is a slower time domain and covers things like the baroreflex. So we don't have a quiz now, but just take a look and see which one you think would have the highest low frequency heart rate variability. So for low frequency heart rate variability, this person actually had the lowest, and here this person had the highest. And what we're looking for low frequency heart rate variability in these plots is some sort of slower oscillation. So like the envelope of this is, you can see sort of going up and down. And here in particular, it's going up and down at what could be, this is 4 minutes. This could be around 10 seconds. So this could be the baroreflex that we're seeing here. So anyway, we have this very strong influence of breathing on heart rate. And much of the good things that have been attributed to heart rate variability are associated with heart rate variability. So it's associated with better mood, less psychopathology. Those signals are driven by how much the heart is responding to breathing. And the reason that the heart does this during breathing is that your heart is tonically being suppressed when you're resting. It's tonically being suppressed, the heart rate, by the vagus nerve, signals from the vagus nerve. So you have pacemaker cells in your heart that would lead it to beat at 100 beats or a little bit higher. And you can take a heart out of the body in transplant situations, connect it to blood flow, and it keeps beating. There are these great movies of this on YouTube, and it beats very steadily. But what's happening when you're resting is that your parasympathetic system is damping that down, putting the brakes on it. And every time you inhale, the vagus nerve transmits signals less effectively than when you exhale. And so that brake is going to be lifted. So if you have a very strong brake on your heart when you're resting, very strong parasympathetic system, you're going to see large oscillations in that in heart rate. So anyway, we can increase those oscillations quite dramatically by slowing down breathing. And this is in part because the stretch signals are going to be larger. There's also stretch receptors in your lungs that are influencing this. Those are going to be larger the more time that you're inhaling and your heart rate is increasing. and then the more time you're exhaling and it's decreasing, these effects are just going to be accentuated. And in particular, if people breathe at around 10 seconds, that's often used in what's called heart rate variability biofeedback, where they found that doing this every day can actually improve anxiety and depression. And we were interested in whether we could intervene to reduce the LC tonic stress-like activity and increase phasic activity with this intervention. So we had an oscillation plus condition where participants breathed at around a 10-second pace and they got biofeedback of their heart rate, and they were trying to increase those oscillations. Another condition tried to keep their heart rate steady using personalized strategies. And we recorded their sessions at home that they did. And here's one example from a participant trying to make their heart rate oscillate compared to their own resting state. You can see they were successful. The condition that was trying to keep their heart rate steady didn't manage to decrease their heart rate variability compared to rest, but they did not increase it, which usually when people are resting, their oscillatory activity of their heart rate increases. So we had these two conditions showing different effects in terms of heart rate oscillations. And because of our question about the LC and its downstream effects on the hippocampus, we were interested in whether we could see differences in structure in the hippocampus, and in particular, those regions of the hippocampus that are strongly innervated by the locus coeruleus. So we created an ROI of 4 of these regions, and in younger adults, there was no change over the 5 weeks of the intervention in structure. In the older adults, they showed relatively increased volume in the oscillation plus group, relatively decreased volume in the oscillation minus group, in a pattern that was associated with the degree to which they increased oscillatory dynamics during their breathing sessions. As I mentioned, noradrenaline is also associated with clearance, brain waste clearance. And so we were interested in trying to get a window into this by taking plasma samples. And we found 2 changes that are consistent with enhanced clearance of waste from the brain. So p-tau 181 is more associated with cerebral spinal fluid, p-tau 181, then total tau is related to cerebral spinal fluid, total tau. So p-tau 181 in plasma is more likely to reflect what's happening in the brain than in the periphery compared to the tau. And likewise for amyloid beta 42/40, which is a widely used ratio reflecting aggregation in the brain, amyloid aggregation, that ratio is in part important because amyloid beta 42 is more aggregative than amyloid beta 40, and it's also more likely to be produced in the brain, whereas amyloid beta 40 is more likely to be produced in the periphery. So as plaques are forming in the brain, this ratio goes down as less amyloid beta 42 makes it to the periphery, is cleared from the brain. And we're seeing that this slow-paced breathing condition is leading to the pattern that would be consistent with greater clearance of these waste products to the periphery. So in summary, we have this small brain region that has this outsized influence, and it has some really interesting features of its anatomy that both have benefits, and then in aging in particular might have some costs. It is the brain's arousal hub region, and it can flexibly enhance processing of whatever has high priority at that moment, which is really important for being able to focus on what you want to focus on at the moment of arousal. And in terms of Alzheimer's disease, it seems to be really important at kicking off the initial phases of the disease. And its structure in healthy adults is associated with better episodic memory. And it's been shown in animal studies to influence clearance of brain waste. And our intervention study suggest that maybe we can intervene to be decreasing tonic levels of LC activity, perhaps also decreasing systemic noradrenaline in ways that might benefit things like brain waste clearance. So very interested in any questions you might have, and just want to acknowledge all of the lab members who led the projects that I talked about today. So thank you very much.
Thank you. It was a very interesting talk. I think we have time for some questions, so don't hesitate just to ask your questions. You can also write them in the chat if you want. Okay. Yes, Nicolas has one. No. Okay, maybe I can start with a question because I was curious about the relationship you showed between phasic and tonic arousal or LC activity. Because according to Aston John's framework, there is like a more inverted U-shape relationship between the two. So are those data like contradictory to this inverted U-shape relationship? Because if lower tonic is associated with the best phasic response.
Oh, I see what you're saying. So you would say, so low tonic would be associated with the best phasic.
Yeah, and for like an inverted U shape, I thought that lower tonic and too high tonic would be associated with low phasic response, and an intermediate would be associated with an optimal response.
Yeah, I think here this is not particularly consistent with a inverted U. I would agree. Yeah. I have difficulty with U shapes in models because usually the evidence isn't very strong. People often will take 2 points and be looking at this. So there's a few data points that do support the inverted U. I think that it's not as strong as it's been made out to be. Yeah.
Okay, thank you. Yeah, and also I have another question about the gain model, because in some cases I think it could be hard to decide if a stimulus is high or low priority. Like in a task, if it's like a distractor stimulus, but it's very salient and unexpected, then how would it be considered in the model? Oh, your sound is deactivated.
Yeah, so I think it's a real tension between what you could think of as top-down, priority and bottom-up priority. So the perceptual salience would be bottom-up. You're not really controlling it. It might be a distractor. And I think absolutely a distractor can have high salience and be more distractible under arousal potentially than something than under not arousal. But there's also top-down goal-directed priority. And that gets pretty complicated because in order to instantiate that priority, in order to say, I really want to pay attention to this letter here that doesn't otherwise stand out, one needs to prefrontal cortex control processes. And actually, arousal tends to diminish that prefrontal cortex control. So under arousal, we have less effective ability to create top-down, to instantiate those top-down goals. So we think probably when it comes to the top-down aspect of, does arousal enhance what you are trying to pay attention to? that will depend on whether you were paying attention to that already. You'd already instituted that goal before the arousing moment happened, in which case that wouldn't be as disrupted by arousal. But if the arousal happens and then your task is to try to pay attention to something that's not all that salient, you probably won't be as good at it. That makes sense. So in terms of the top-down aspect, I think there's some influence of the timing of when you would be trying to make that salient yourself. So the perceptual salience, I think, distracting information, that will still, that salience will come through under arousal at a higher level.
Okay, thank you. I think Aurélie has a question.
Sorry, my question. Thank you so much for the presentation. I have a naive question. So I know you're a specialist on aging, but I was wondering if you know anything about development during childhood, if the locus ceruleus, when is the locus ceruleus mature, and if there is a different level in tonic arousal across childhood, and that could also have a link with the difference in distractibility we can observe in younger children.
Yes, this is a great question, and one that has not gotten much attention at all. The locus coeruleus does seem to develop quite early, just in terms of, having the ability to function and having this noradrenergic release. We don't know much about how it relates to, attention deficit disorder, attentional deficits. It seems like it would. I mean, certainly the drug regimes that kids with attention deficit disorder are put on influence the noradrenergic system. We are excited because we actually are just starting to look at this in a large data set where there is some imaging available that we can look at the MRI contrast, and we can look at some attentional measures. So you can stay tuned. Martin Dahl is working on this, and so we will be able to start, this is, not the same as being able to look at LC activity, but in the aging side, we've seen that it really can be quite interesting and informative to just look at this structural measure of the LC. And so we should be able to start doing that with some of these data sets that have been collected with kids that have different measures as well.
Thank you. I have another question, if I may.
Mm-hmm.
I was wondering, do you have any opinion about indirect measures of the locus coeruleus activity in humans, such as skin conductance or pupil dilation?
Do you mind repeating it? I didn't catch all of it. Yeah.
I was wondering if you have any opinion about indirect measures of locus coeruleus activity in humans, such as pupil dilation or skin conductance or heart rate.
Yeah. So of those, I would say pupil dilation has the closest link or most specific link to the locus coeruleus, although it is not entirely specific. There are other brain nuclei that also lead to pupil dilation. But we do know from very nice studies that have been done that stimulating the locus coeruleus does lead to pupil dilation. And also the P300 is another one that, ERP measure that's been widely used and also been documented to, stimulation of the locus coeruleus leads to increased P300. Now, doesn't mean that P300 necessarily means that the locus coeruleus has activated. So you have to be careful in interpreting it, but I think it's really useful because it does provide a window, and you can be inferring some of these processes from it. But it's not a one-to-one mapping, so it's not going to be perfect. Skin conductance and heart rate are influenced more by more broad systems. So they're not usually used directly to infer anything about the locus coeruleus, just more broad arousal system specs.
Okay. Thank you.
Yeah, with pupil dilation, we've seen this sort of tonic phasic effect with hand grip, where we see greater, just tonically higher pupil dilation, larger pupils, restricts the phasic response to an oddball sound. So it looks like what we would predict in terms of LC activity. There's also interesting dynamics in terms of if after hand grip, we would see that there was less, so less pupil, smaller pupil size that seemed to perhaps reflect a depletion of noradrenaline. So there's some level at which if you stimulate the locus coeruleus quite intensely, you can deplete noradrenaline briefly, and you won't see as big effects after that. So the phasic tonic relationship, actually, they antagonize each other. Not only does tonic prevent phasic, but phasic prevents tonic in the immediate aftereffect. So if you have a large burst of LC activity, it somewhat perhaps depletes noradrenaline. Basically, a large burst of LC activity, you get less tonic activity afterwards.
Thank you.
There is a question in the chat, so maybe I can read it. Yes, so could we imagine the same patterns of curve with the inversion of sympathetic and parasympathetic activities during day and night in people with sleep disorders? And how could this be related with the impairment of attention?
Yeah, so absolutely. So people with sleep disorders, definitely the sympathetic system gets hyperactive. So this is especially seen in people with sleep apnea, where, there's breathing obstructions that lead to arousal, that leads to awakening. And that leads to hyperactive sympathetic activity throughout the day in a, pretty dangerous sort of risky situation. It seems to be part of the health risks of sleep apnea. And yeah, I think that this could be related to impairment of attention. If the system is, first of all, of course, the person's going to be more tired, and that's probably the biggest influence. But if these systems are not operating as effectively, if there's high tonic levels of noradrenaline, the locus coeruleus is hyperactive, then the phasic responses won't work as well, and that can be problematic in people with sleep disorders.
Okay, thank you. There is another question in the chat. So you mentioned a potential link of baroreceptor sensitivity and LC activity. What would you expect would be the link of blood pressure variability and LC activity or LC integration?
Oh, this is a great question to think about this. So colleagues of mine at USC, Dan Nation and his lab have been looking at blood pressure variability. So blood pressure variability is the opposite of heart rate variability in terms of its whether it's considered good or bad, right? So heart rate variability, higher is better. It's indicating an effective parasympathetic system that is effectively suppressing your tonic heart rate and responding to breathing. Blood pressure variability is bad generally. It is reflecting a dysfunctional baroreflex that's not keeping blood pressure tightly regulated. So blood pressure variability has been looked at in various types of ways. So the simplest way is just visit to visit differences in blood pressure. So if you're an older adult and some visits you have high blood pressure, some low. That's not a good thing. It's associated with bad outcomes. Also, just blood pressure variability within a session. And so in terms of the locus coeruleus activity and links with blood pressure variability. So the locus coeruleus is, just one, the noradrenergic system, sympathetic system is just one piece of this baroreflex loop. And it could be that when that gets less effective, you would see more blood pressure variability if it's not responding as effectively. But of course, it could also be the vagus nerve parts of it. So it certainly could be related. I don't know that we have evidence that would point to it right now, but it's a really interesting question.
Thank you. Are there any more questions? No, maybe not. So if there is no more question, I think we can move on to the part only with the PhD students, so we can discuss. So PhD students, you can stay. And the other, well, thank you for attending the talk.